Okay, so in this video, I'm going to be talking about multiple things, but I'm going to start off by talking about homeostasis. Homeostasis is the body's ability to regulate its temperature, pH. A lot of different things that happen inside of our bodies undergoes homeostasis. How how I'd like to think about this is by drawing a graph. Homeostasis is if this line was the perfect, let's say body temperature, we'll start with that. If this line was the perfect body temperature, homeostasis would be keeping it within a normal range. It means keeping it here. So it might go above and below a little bit, but it's in the normal range. Occasionally, it might go outside the normal range, and that's usually bad. There's a few instances where it might be okay, but um, keeping it inside the normal range is what usually our body does. So let's go back to body temperature, and we'll go a little bit more in detail. Um, let's say we have some sort of stimulus. This means that the outside environment changed, and now our body needs to react to maintain our body temperature. This could be like a change in temperature, it's cold outside or something like that. What our body does is there's this stimulus, and then we'll have a receptor. And in this case, we'll have thermoreceptors all over our body that will tell us what the temperature is, and then we'll send that signal to a control center. So in this case, it would be the hypothalamus, which is in charge of the temperature portion of our brain. Um, then that's the afferent pathway sending that signal to the control center. The efferent pathway is what the control center sends out to the effectors. The effectors are the things that actually will put it back to the set point, and keep it within this normal range. That's what our effector does, and the control center sends signals to the effectors to do their job. So in the example of our temperature, it could be sweat glands, or it could be um, our blood vessels dilating, or something like that to keep our temperature controlled. What we'd see here, we have a stimulus. We're going along, then we have a stimulus. I'll use a different color for this. We're going along, and then we have a stimulus that will raise our body temperature, or we'll say lower. Keep it the same. It'll lower our body temperature. Then it hits the control center, and we send an effector. We'll say sweat glands happens, and it gives us back to our set point. That's what we call a negative feedback loop. And for negative feedback loops, it is anytime we send something back to its set point, or send that stimulus back to the set point. The opposite of that, the positive feedback loop, is when we send something away from the set point. This usually means that the effectors are not doing its job correctly. So let's say we're going along, we hit that same stimulus, but then there's something wrong with the effectors, and it does the opposite. Then we go outside of this normal range here, and this is a positive feedback loop. There's occasions where this is good. For sometimes during childbirth, this happens, and it's okay. But most of the time, we want to keep within this normal range. Okay, so that is homeostasis and feedback loops, all that good stuff. Next thing I want to talk about is the protein-mediated transport. We have a few different kinds of protein-mediated transport, including we'll talk about simple diffusion. We'll talk about facilitated diffusion, and we'll talk about positive primary and secondary active transport. So let's start with simple diffusion. I'm going to draw a cell membrane here. Diffusion is anytime we have something traveling through the membrane. Simple diffusion means that we have something out here that wants to come in here, and it's small enough and that it can travel through. There's a few other properties, like its polarity will affect this as well, or its charge will affect this, but it needs to have certain criteria, and then it'll travel into the cell without any help. This is passive transport, so it doesn't require any ATP. But let's draw a channel here. So this is a channel. It's very specific. Only certain things can travel through these channels because, like I said, there is criteria to be able to travel through a cell membrane so easily. For instance, CO2 that can travel through the membrane pretty easily. Let's say we have a lot of CO2 out here and only a little bit inside the cell. What happens here is this CO2 wants to get inside the cell because it wants to even out its concentration gradient is the term. It will follow its concentration gradient until it's at equilibrium. So we have one CO2 will come in, and then it's at equilibrium. It's usually not that even because there's much more than 4 CO2s in our body. But you get the picture. That's simple diffusion, requires no ATP. It is specific, and it's very fast because it requires no help. Okay. Next, we'll talk about facilitated diffusion. Facilitated diffusion is when we have a little bit of help through proteins. There's something that are usually a little bit bigger. Things that can't just go through the membrane, but they need help doing it. For instance, we might have sodium. We might have sodium come through. Let's just use that example. Okay, so we have a lot of sodium outside, and we are trying to get more sodium inside the cell for equilibrium. There's multiple different ways that facilitated diffusion can occur. These can be different kind of channels. It is a protein that's involved. That's this blue structure here, but there needs to have some sort of reaction or something needs to happen to this protein for it to be able to transport a sodium across into the cell membrane. There are different ways that this can do this. Sometimes it's a ligand, so a smaller protein will come and attach itself. Sometimes it happens through mechanical means, so the actual cell membrane will change shape, and that will signal to the protein to let a sodium inside. Or voltage, so there would be a charge of some sort that signals to the sodium, that signals to the protein to let sodium inside. But something needs to happen to this protein to signal that we're ready to go inside. So the sodium will attach, and then some sort of signal will happen, and a conformational change will happen to this protein and send the sodium inside. Like I said, this is when I was describing it. This is also passive. There's no ATP required for this because it will only occur when it's just binding things like the ligand or mechanical or voltage. These don't require ATP. It's just a signal for the protein to move and change. These are very high specificity because the sodium or whatever it is needs to actually bind to the protein. And so there's these binding sites that they need to match up. But this also goes from a high to low concentration. This is trying to maintain some sort of equilibrium. Okay, lastly, we're going to talk about primary and secondary active transport. Primary and secondary active transport is a little bit different. So we're going to have a protein. These proteins are usually a little bit more complicated than just undergoing some sort of conformational change. But we're going to use an example of the sodium and potassium ATPase pump. That's going to be primary transport. Primary transport means that we are directly using ATP. So, we're going to use the sodium example again, and we're also going to bring in a potassium. And then we're going to have a few more potassiums inside the cell. Okay, so the sodium and potassium ATPase pump, what it will do is it'll actually go on the it'll actually go against its concentration gradient. Here, I'm gonna add some more. So what will happen is it will send sodiums. It'll send sodium. Let me check something. So what's going to happen here is we're actually going to go against the concentration gradient. So what we're going to have is we're going to have 3 sodiums. This is what the sodium potassium ATPase pump does. We're having 3 sodiums leave the cell, and two potassiums come into the cell. As you can tell, this is different. The concentration gradient, there's more sodiums inside the cell, and there's more sodiums outside the cell and potassiums inside the cell. So this is going against the concentration gradient. That means we have ATP coming in. ATP will give it the energy that it needs to go against this concentration gradient. That's primary active transport. Secondary active transport is going to use this for its advantage. For instance, we'll also have a glucose symporter. So we have a lot more extra sodiums outside. So these sodiums, they really want to get inside now. So over here we'll have a glucose symporter. So we have sodiums trying to equal out their concentration gradient. But then we'll also have glucose that will use this opportunity to travel inside the cell without any energy. Because sodium's just trying to get across to equal out its concentration gradient. So glucose is going to take advantage of that and equal things out. So that's secondary active transport. This again is from low to high concentration. Because this is again is from low to high concentration because it will. This again is against its concentration gradient because glucose here is going against its concentration gradient. So some sort of energy is needed, and so the energy from sodium is what's being used. So these two things do require ATP. This one uses it directly. This one, it takes advantage of the ATP used here. Okay, sorry, the video cut out there a little bit. I'm going to talk about a little bit on osmolarity, osmosis, and tonicity. Osmolarity is the measurement of particles, measure of the number of moles of particles in a solution, and molarity is the measurement of the number of molecules in a solution. So there could be a little bit of a difference there. The osmosis is the measurement of water entering another the measure of movement of water from low solute concentration to high. So let's say we have a cell within a beaker, some sort of solution. If we have, here's our solute. We're going to say there's 4 particles there. That's not going to happen, but that's what we'll say. Inside the cell, there's 6 particles of solute. If we're measuring osmosis, water is going to move from low solute concentration to high into the cell. That's osmosis, is movement of water. Now, tonicity is the effect that movement of water has on the cell volume. So, osmosis means that we're having movement of water, and then tonicity is the effect that has on the cell volume. So, in this example, we're comparing these to this. We're comparing the solution to the cell. The solution is going to be hypertonic, meaning that we are losing. Meaning that we are losing water from the solution. And then hypotonic, the cell is hypotonic because it's gaining. It's hypoosmolar because it's gaining water. Now, in that case, we are also having this. The cell is reflective of is hypotonic as well because we have more. Of the more water entering this cell, and it is going to expand and become hypotonic. Well, that is tonicity and osmolarity as well as osmosis. That's all I have.